What if I told you that the most dangerous accomplice in a breast cancer tumor isn't actually a mutated cell at all? It's the healthy fat tissue you've had your entire life. We tend to visualize cancer like a spy movie. There's your healthy tissue, just doing its job, and then one rogue agent mutates, multiplies out of control, and the mission for the good guys is to find that lone villain and take him out. It's a clean narrative. It separates the body into a binary, the villain and the innocent background tissue. But when you step into the actual research, specifically the area scientists call the tumour microenvironment, that clean narrative shatters. Cancer isn't just a rogue cell acting alone. It acts like a corrupt neighbor that actively brainwashes the surrounding tissue to do its dirty work. The creation of what researchers call cancer-associated adipocytes, hijacked fat cells, is arguably one of the most underappreciated stories in modern biology. To understand the hijacking, we first have to understand the hostage, normal fat. For most of the history of cancer biology, the fat surrounding a tumor was practically invisible. When pathologists prepared tissue samples using standard chemical staining, the solvents used literally dissolved the fat right out of the tissue. Researchers looked at breast cancer slides and saw only empty white spaces where the fat had been. It was treated as bubble wrap. Background padding, blank space. But once scientists figured out how to study fat without destroying it, they found something remarkable. A healthy fat cell, an adipocyte, is not passive at all. Picture a water balloon. A single enormous droplet of stored fat dominates the interior, pushing all the cellular machinery flat against the outer wall. It looks like a storage locker, but it's actually a bustling chemical factory. Fat cells are constantly secreting a sophisticated mixture of hormones and signaling molecules called adipokines. They regulate insulin response, they supply nutrients to surrounding cells, they communicate with the immune system. In breast tissue, which is predominantly fat, the surrounding adipocytes maintain a self-regulating, healthy ecosystem for the milk duct cells where most breast cancer. So when a rogue cancer cell begins mutating inside a milk duct and starts pushing outward, it isn't landing in random padding. It's landing right next door to a fully stocked automated chemical factory, and it immediately sets about taking it over. The hijacking begins at a distance. Before the tumor even physically touches the fat cell, it sends out signals through the fluid-filled space between cells. The first delivery mechanism is exosomes, microscopic membrane-wrapped parcels. Think of them like messages in a bottle. The cancer cell packs these exosomes with corrupted genetic instructions, specifically microRNAs, and releases millions of them into the surrounding fluid. When a healthy fat cell absorbs one, it reads the instructions: shut down your master identity genes. The cancer also secretes WNT proteins directly into the fluid. These drift over and dock onto fat cell receptors, triggering a chemical cascade that suppresses the genes required to maintain the cell's normal identity. By the time the cancer physically arrives, the fat cells have already been reprogrammed to serve it. The reprogrammed fat cell undergoes a radical transformation. That plump water balloon deflates. The single massive lipid droplet shatters into chaotic tiny fragments. Under an electron microscope, the hijacked fat cell looks wrinkled and exhausted, a shadow of its former self, and its job description has been completely rewritten. A normal fat cell produces two key hormones locally, leptin and adiponectin. In healthy tissue, adiponectin acts as a natural brake, suppressing unchecked cell division and encouraging abnormal cells to die. But once hijacked, the fat cell floods the environment with leptin, which acts as an aggressive growth factor, pushing cancer cells to divide and invade. Meanwhile, adiponectin production plummets. The gas pedal is floored. The brake lines are cut. The hijacked fat cell also releases a flood of inflammatory signals, chemokines like CCL2, that lure macrophages, the immune system's cleanup crew, directly into the tumor environment. But instead of attacking the cancer, those macrophages are chemically converted into a pro-tumor state. They start building blood vessels and structural scaffolding for the tumor to grow on. The immune system's own soldiers are recruited to construct the enemy's infrastructure. To physically escape the milk duct, the tumor needs to dissolve the collagen barrier, the basement membrane that contains it. Hijacked fat cells do exactly this, releasing enzymes called matrix metalloproteinases that act like molecular scissors, chewing through the collagen wall. Then they alter the wider collagen structure, making it stiffer. Counterintuitively, this stiffness is exactly what the cancer wants. When a cancer cell feels that physical tension, it triggers a genetic response that makes it more aggressive and more invasive. Pathologists can literally map collagen stiffness in tumor samples and predict how invasive the cancer will be. The tumor also needs fuel, enormous amounts of it. Hijacked fat cells break down their stored fat reserves into free fatty acids and pump them directly into the surrounding fluid. Cancer cells have upregulated import proteins on their surface, acting as biological funnels to pull those fatty acids in and burn them for energy. The cancer orders takeout. The fat cell breaks down its own body weight to cook and deliver it. For hormone receptor-positive breast cancers, over 2/3 of all cases, the most devastating part of this partnership is the estrogen loop. After menopause, fat tissue becomes the body's primary estrogen producer via an enzyme called aromatase. Hijacked fat cells are forced to massively ramp up aromatase production, flooding the local environment with estrogen that feeds directly onto the cancer cells. As the tumor grows, it corrupts more fat cells, which make more aromatase, which makes more estrogen. A postmenopausal woman may have very low systemic estrogen, but inside her breast tissue, the tumor has built its own private hormone factory, with local concentrations up to 10 times higher than the rest of her body. And the hijacked fat actively shields the tumor from treatment. It suppresses the immune system's killer T cells. It paralyzes natural killer cells. It acts as a literal sponge for immunotherapy drugs, binding to the antibodies before they ever reach the cancer. It upregulates efflux pumps that physically eject chemotherapy molecules, and it overwhelms aromatase inhibitors through sheer volume. The tumor simply corrupts enough fat cells to outproduce any drug blockade. Now here's where the story becomes genuinely empowering. When a person carries excess adipose tissue, it often exists in a pre-inflamed state, already producing elevated leptin, reduced adiponectin, higher baseline aromatase. The microenvironment is pre-tilled soil. When cancer arrives, it skips the heavy lifting and goes straight to exploitation. This is the specific biological mechanism behind why obesity is an independent risk factor for worse breast cancer outcomes. But understanding the mechanism transforms the prescription. Exercise and tissue health are no longer vague wellness advice. They are targeted biological interventions. When you reduce the volume and inflammatory state of your adipose tissue, you lower local leptin, raise adiponectin, and dial down aromatase. You are literally starving the tumor's supply chain and making the neighborhood hostile to invasion. Researchers are now building weapons that target this partnership directly. Metformin, the frontline diabetes drug, acts as a firewall in laboratory models, preventing fat cells from being fully hijacked. Drugs targeting leptin receptors would deafen the tumor to its own growth signal. Synthetic adiponectin mimics could restore the biological brakes. And anti-inflammatory drugs like canakinumab showed striking reductions in cancer mortality in clinical trials, simply by making the tissue environment less welcoming to tumors. We are no longer just trying to bomb the rogue cell. We are attacking the supply lines, jamming communications, and unbrainwashing the accomplices. Cancer is not a lone villain. It is an ecological crisis, but systems can be understood, and systems can be disrupted. Fat is not bubble wrap. It is not a bystander. It is an active, communicative organ, one that can be manipulated by disease, but also one that can be understood, targeted, and ultimately turned back against the tumor. Every biological mechanism you understand is a new tool in your toolkit. Stay curious.